Hi everyone, it's Mr. Sinti, and today I'd like to discuss with you today one of the more serious and important topics in molecular biology, and that is how the cell cycle is regulated. And when that regulation normally occurs, things are great, but when there are some abnormal events that occur during cell cycle regulation, it can result in something pretty serious, which is the formation of cancer. Now, I know that this is a serious topic and it requires serious attention, therefore. And hopefully this video, it's a brief look actually, it's not going to go too far into it, but I hope it stimulates you, more importantly than just understanding the content. I hope it stimulates in you a desire to want to research it even further. That goes for all the things that we're really talking about, but this in particular, cancer is a killer and it must be stopped. And I think it begins with the awareness and the motivation to do so. So one of the things that I want to talk about is that gene regulation systems can go wrong. And when that happens, it can result in abnormal cell growth, which is cancer. And so as you can see, a cancer here is an outgrowth of cells in the back of the retina of an eye. This is an example of a cancer, the cancer of the retina called retinoblastoma. So I'll just give you an example of that. So there's genes associated with cancer. And so just to establish that, the mutations, which are changes to nucleotide sequences of DNA, can affect whether or not the cell properly regulates growth and division. And some of the things that can affect this is mutations in genes having to do with growth factors. You may be familiar with the fact that growth factors initiate cells to know when to grow or when not to grow. The mutations could occur in proteins that are found in the membrane of cells that receive those signals from the outside, or it could be mutations in the signal transduction pathway leading to cell division, or it could be in a number of genes that are responsible for tumor suppression. And so that's what this video is all about. And so I also wanted to point out something that you may or may not be familiar with, that viruses, which are small proteins surrounding DNA or RNA that are not living, so these biological particles, some viruses are tumor viruses, and they're known to cause cancer. You might be familiar with this. Like things like hepatitis B virus can lead to liver cancer, and the papilloma virus can lead to cervical cancer, and Epstein-Barr virus can lead to mononucleosis. Usually though, viruses can cause it, but sometimes we think about the things that cause mutations or cause damage to DNA as being ultraviolet light or carcinogens, which are chemicals that are unfortunately present in our environment, and sadly some of them are man-made. And so just to establish some basic terminology so we can talk about this more profoundly, oncogenes, that's an important term. Oncogenes are cancer-causing genes. And the study of cancer is oncology, so oncogenes. So proto, meaning coming before, proto-oncogenes, are the normal cellular genes that are responsible for cell growth and division. That's what we're talking about here, cell growth and division in particular. It's the conversion of the proto-oncogene to an oncogene that leads to the abnormal stimulation of the cell cycle and thus responsible for initiating cancer. You might be familiar with the cell cycle. There's interphase, and then there's the mitotic phase, which mitosis precedes cell division, which is cytokinesis. Okay, and there's a few checkpoints along in the cell cycle. You might be familiar with this. I actually produced a video on the cell cycle regulation. You're welcome to check that out. But there's an important checkpoint right here at the beginning of DNA replication. The cell sort of assesses how things are going in terms of its growth, whether or not it's ready to replicate its DNA or not. And so there's a couple of proteins, and I won't go into the details of this, that regulate the the process of DNA synthesis and regulate whether or not a cell undergoes mitosis or not. And these are called cyclin-dependent kinases, or CDKs. You may recall these proteins somewhere in your past. Cyclic-dependent kinases. And what they do, very briefly, is that they'll add a phosphate to various proteins. And what that'll do is either activate or inactivate proteins. And so these CDKs, as you might imagine, work together with cyclins. Okay, so they're cyclin-dependent kinases, and they work in concert and bind with cyclin, and that's how they do their work. So the first checkpoint, as I was mentioning, is right over here, right in front of DNA replication. So there's certain cyclins, and thus they cycle. Certain cyclins are made at different times in the cell cycle. And the main point here is that only when the CDKs are bound to the cyclin that they become active. And so as you can see in this diagram down below here, that cyclin D are produced early on in G1 and cyclin E as well, and then the initiation of cyclin A starts to happen here. And so you'll see that these cyclin Ds and Es are getting produced early in G1, and then they bind to kinases. And cyclin-dependent 4 in particular will phosphorylate a protein called RB, which is going to be important in a moment. I'll discuss that. And so what RB does is that normally RB, this protein, inhibits DNA replication. That's what it does normally. However, when RB has a phosphate stuck on it, it becomes inactive, and thus replication can proceed. If you're following that, let me repeat that. So it normally inhibits replication, but when it has a phosphate stuck on it, it becomes inactive, it changes shape, and so therefore replication can proceed. Okay, so as we move through the S phase, you can see that cyclin A is being made, and it's going to complex, and it binds to cyclin-dependent 2, and that activates DNA replication. Then as you move along, cyclin B, that's produced during G2 phase, binds with cyclin-dependent 1 and activates cell division. So just to review all of that, you may want to pause and take notes if necessary, but so just to review the process, through these checkpoints, you need to have cyclin proteins present in order to move through the cell cycle normally. So the control, here's a main point, the control of the cell cycle occurs on a higher level though. So one of the key molecules in regulation of the cell cycle is a protein called P53, and that might be kind of random, but it's actually named after the fact that the protein in question, P53, is 53,000 Daltons molecular weight. It's a pretty significant protein, as it turns out, and it's actually referred to somewhat appropriately as the guardian angel of the genome, meaning that it's really crucial and it sort of watches over things. As it turns out, it's a transcription factor. You might be familiar with those. Those are necessary for RNA polymerase to effectively bind to DNA. So when it binds to DNA directly, it then allows the DNA to produce a protein that blocks the progression of the cell cycle. Okay, and one of those proteins that does this is P21. And again, there's a gene for P21, but it's the protein that I'm talking about. Its function is to inhibit cyclin-dependent kinase. And so it will not be able to activate DNA replication or activate mitosis. So in other words, RB, which is another protein that can inhibit DNA replication. So these proteins, if you will, these molecules are then considered to be tumor suppressant, and the genes that produce them are considered to be tumor suppressant genes because their activity prevents DNA replication and mitosis from occurring. So the thing is, if these tumor suppressant genes have a mutation, they can't function properly, and it results in the cells having abnormal cell cycle or cancer. Okay, and so how important, you might ask, is this? Well, about 50% of tumors have defective P53 genes. That's significant. Likewise, RB, which gets its name from retinoblastoma, as I was referring to at the top of the video, as well. And so some tumor suppressant genes code for proteins that repair DNA. If you're familiar with the fact that sometimes ultraviolet light damage can cause thymine dimers in the DNA, and then through an excision repair process, that portion of the DNA is cut with endonucleases, and then it's repaired by an RNA polymerase. Some tumor suppressant genes that repair DNA, the function is not working, and so therefore mutations accumulate because those proteins are not available. And so this is an age-dependent phenomenon. In other words, as a person gets older, they're more likely to occur more mutations over the course of a lifetime, and that will result in cancer. So the idea is that these genes are pretty important, and they make sure that the cells divide and don't divide, and in particular, control the cell cycle. So they're pretty important. And so what's going on? Like, how can these proto-oncogenes be converted to oncogenes? Well, here's just 3 ways in which this can happen. You can have movement of DNA. DNA is capable of, pieces of DNA can actually excise from one place on a chromosome and actually relocate. It's basically like moving from one place in a city to another place. You can move around, and when DNA moves around like that, that's kind of haphazard. It could end up near, if it's a crucial gene, it can end up near an active promoter, and that increases transcription, which may influence cell cycle regulation. Or there could be simply repeats. There can be amplification of particular genes, and they're copied more than other genes. Or there could be simply a point mutation, a single nucleotide substitution, base substitution in a particular control element, which causes an increase or decrease depending on what activator protein attaches to it and transcription factors. Now, I know that's kind of a lot to take in, but here's a diagram of this. So translocation, in other words, a proto-oncogene actually moves into a new location, and there's a new promoter, and that stimulates abnormal production of or excess of a protein. And as I mentioned before, you can have gene amplifications, or many copies of the genes, or repeats. And of course, that's going to influence the overproduction of a protein, which would affect the cell cycle and thus possibly result in an oncogene, which then results in cancer. Or you could have a point mutation. Like, for example, here's the gene, and then this line suggests that this is pretty distal from the location of the gene. But there could be a point mutation within a control element. In other words, where proteins bind to this. And so you can have over stimulation, and so hyper activity or degradation of a resistant protein. You can have problems with the result of mutation, gene amplification, or translocation. And so tumor suppressant genes are really important because simply, and let me emphasize this. Whoops, let me go back over here to. Didn't mean to push that. Sorry about that. Here we go. Whoops. Tumor suppressant genes are really important because they help prevent, thus the name suppressant, they help to prevent uncontrolled cell growth. So that's rather important. And so mutations that decrease protein production of tumor suppressant genes result in cancer onset. So these are highly studied areas in molecular biology and oncology. And so what some of these tumor suppressant proteins do, as I alluded to them a little bit earlier, is that they repair DNA. That's one of their functions. And they also regulate the cell cycle. I've been talking about that. But they also, in fact, affect whether or not a cell is adhering to a particular substrate or other cells. I won't get too far into this, but cell adhesion is a way in which cells recognize that there's crowding going on in tissue and therefore stop growing. And so when cells are not able to recognize that, they become cancerous. And without going into a lot of detail here, and this is the heart and soul, I think, of cancer research currently, is that there's different stages within signal transduction pathways that occur. So it's not just abnormal receptor proteins, but it's the proteins or the secondary molecules involved in signal transduction that leads to regulation problems in cancer. And so let's talk about a few examples of this. So a mutation in a RAS proto-oncogene. Okay, so RAS is rat sarcoma. Proto-oncogene and p53 tumor-suppressor genes are common in human cancers. So these areas are highly studied. So a mutation in the RAS gene can lead to hyperproduction of RAS proteins and increased cell division. And so the RAS protein, if you're not familiar with this, is a classic G protein, which is involved, as I was mentioning before, in signal transduction pathway. So again, you could have abnormal receptors that will receive more signals to divide growth factors. And then you can have this RAS protein. What's interesting is that it'll actually initiate a signal transduction pathway when it doesn't even receive a message from the outside. And as a result, it produces and stimulates the cell cycle to be hyperactive, and therefore the cells divide uncontrolled. Okay, and so you can also have a problem with the p53 as well. And so that protein normally inhibits the cell cycle. And so if there's a mutation in that, it can't activate transcription, and therefore it doesn't prevent the cell cycle from happening, and so therefore you have uncontrolled. So mutation in the p53 prevents the suppression of the cell cycle, thus cancer. Okay, if that makes sense. So mutation to that gene and the RAS gene is significant. And again, the effects of mutation are in general affecting proteins that regulate the cell cycle. So if there's proteins present that should be, or if there's proteins not present that should be, it's ultimately going to affect the cell cycle. And so this process sometimes, fortunately, is when cancer actually develops, it's usually a multiple step model. Generally, there needs to be multiple mutations for full-fledged cancer to occur. And so this, again, accumulates usually over the course of a person's lifetime as they get older. And as DNA level, the cancerous cell usually is characterized by more than one active oncogene. And so they start to accumulate mutations in several tumor-suppressor genes, and then ultimately the individual succumbs to cancer. And one of the more troublesome cancers is colon cancer. You might be familiar with the colon, which is the large intestine. Its function is to reabsorb water mainly. The lining of the epithelium layer of the colon can become tumorous. Here's a malignant tumor, carcinoma, stage 4, meaning that it's going to metastasize and spread into the lymph nodes surrounding it, which is potentially lethal. You can inherit these mutations that are passed from your parents. And so individuals that have mutations in the gene in particular that I'm talking about with regard to colon cancer is APC. And so you could do a genetic screen and determine whether or not you're a carrier for this or homozygous for this, heterozygous or homozygous for this. And so that's fairly hopeful, because you might be able to then go in for more colonoscopies, or you could take precaution if possible. And then finally, I wanted to talk about breast cancer, which is a real devastating disorder. 1 in 8 U.S. women, about 12% of the population, will eventually develop this invasive breast cancer over the course of a lifetime. And so for women in the U.S., breast cancer rates are higher, can you believe this, than any other cancer besides lung cancer. And so it's a problem that affects, you probably are familiar with someone who has breast cancer. which is unfortunate. And so it's, again, I said this at the top of the video, and sorry for coming in so strong and with this statement, but it's, you know, the awareness, the fact this, it's gotta motivate us. This is a social awareness which needs to impel us to action, because the fact that 5 to 10% of the cancer is linked to abnormal mutations in the genes, and the fact that it's inherited, makes us want to rally and look and find these genes. And so we have located some of these genes. So a mutation in the BRCA1 and 2 genes are some of the genes that are most common in inheritable breast cancer. So on average, women with this BRCA1 mutation have approximately a 60% chance in the lifetime of developing breast cancer. Now, that's important information to have. So if you had your gene screened and you had that mutation, you might want to take a preventative measure. In other words, you might want to go in for a mammogram more often, or you might even want to take the action. I'm not a physician on this, but you may want to take the action of removing the breast tissue in order to prevent that from occurring. Okay, so a mutation here is a 45% chance of developing breast cancer, and so these mutations are found in at least half the inherited breast cancers. And the way you can do that is simply to sample your cells. You can amplify your these particular genes using primers, using PCR, and then you would run sequence on that to determine whether or not you had a mutation in those genes. And so it's something. The first step is knowing that you have the gene in the first place. So I hope this particular video shined a little bit of a light on how genetic changes can affect the cell cycle control. All right, thanks for watching.